Right. We go for the next one, that is the parts of the nephron, and they are lined by and permeable to and impermeable to. For example, you take the first one, the Bowman's capsule is lined by simple squamous epithelium, and proximal convoluted tubule is lined by simple cuboidal epithelium with a brush border. That means the cuboidal cells consist of microvilli. Due to presence of microvilli, it gives a brush border appearance. So the proximal convoluted tubule is permeable to both water and electrolytes. The next one is the descending limb of loop of Henle. It is lined by simple squamous epithelium and it is permeable to the water and nearly impermeable to the electrolytes. Then ascending limb, if we take it is lined by simple squamous epithelium. That means in the ascending limb, there are two segments. One is the thin segment, one is the thick segment. Thin segment is lined by simple squamous epithelium and thick segment is lined by simple cuboidal epithelium. Both the thin and thick segments are permeable to the electrolytes and impermeable to the water. Right. Next one is the distal convoluted tubule. Distal convoluted tubule is lined by simple cuboidal epithelium, but the microvilli are absent. So due to absence of microvilli, we can say that the brush borders are absent. And distal convoluted tubule is permeable to water and electrolytes. Next one is collecting duct. Collecting duct is lined by simple cuboidal epithelium and it is permeable to the water and electrolytes. So let us see the diagrammatic representation. So this one is the Bowman's capsule. This is the PCT. This is the descending limb. This one is the ascending limb. And this one is DCT. And this one is the collecting duct. So here if you see with the red color, so it is the network of blood capillaries, which I'm covering with the black lines now. The network of blood capillaries present around the PCT and DCT is called as peritubular network. So the network of blood capillaries that surrounds the proximal convoluted tubule and distal convoluted tubule is called as peritubular network. Then the network of blood capillaries that surrounds the loop of Henle is called as vasa recta. The network of blood capillaries that are surrounding the loop of Henle is called as vasa recta. Okay. Then if you see all the collecting ducts, all the distal convoluting tubules of the nephron, that means if you take the one renal pyramid, in one renal pyramid there are many nephrons. So all the DCT of our nephrons are opening into a straight collecting duct. And all these collecting duct opens into the duct of Bellini. Duct of Bellini opens at the tip of renal papillae, and renal papillae opens into the calyces. Okay, first one is minor calyces. Minor calyces opens into major calyces, and major calyces opens into the urethra. So what are the parts present in the cortex? In the cortex, we can see Bowman's capsule, proximal convoluted tubule, and distal convoluted tubule. And in medulla, we can see only the presence of loop of Henle, that is the descending limb and ascending limb, and also collecting duct. Collecting duct also extends into the medulla. Now let us see the conducting system. Conducting system means from where to where the primary urine or renal filtrate is flowing. Okay. So we see here the distal convoluted tubules of many nephrons opens into the collecting duct. All the collecting ducts opens into ducts of Bellini. Duct of Bellini opens into renal papillae. Renal papillae into minor calyces. Minor calyces into major calyces. Major calyces into renal pelvis. Renal pelvis into ureter. Ureter opens into urinary bladder, and urinary bladder opens into urethra, and urethra opens outside through urethral meters. In the previous diagram, you have seen all these parts, right? Now let us see how the blood supply. This one also I explained in the diagram. So renal artery opens into afferent arteriole. Afferent arteriole forms the network of blood capillaries in the glomerulus. In the Bowman's capsule and forms the glomerulus. Glomerulus opens outside through efferent arteriole. Efferent arteriole forms the peritubular network around PCT, DCT. And from peritubular network, vasa recta arises, and vasa recta opens into renal venule, and renal venule opens into the renal vein. So all these points are very, very important for the NEET purposes. Now let us see the formation of urine. So urine formation occurs in 3 stages. Okay, formation of urine occurs in the 3 stages. What are those 3 stages means? The first one is glomerular filtration. Second one is selective reabsorption. Third one is tubular secretion. Let us see the first one, glomerular filtration. Glomerular filtration is also having another name called as ultrafiltration. Why we are calling this one as ultrafiltration means glomerular filtration occurs due to pressure. As it occurs due to high blood pressure, it is called as ultrafiltration. Where does the glomerular filtration occur? The glomerular filtration occurs in Bowman's capsule, and the glomerular filtration occurs through filtration membrane. So glomerular filtration occurs due to the high blood pressure. You know that when the blood is flowing into the glomerulus, it flows with some blood pressure. So the blood pressure that is entering into the glomerulus is called as glomerular hydrostatic pressure. This glomerular hydrostatic pressure is opposed by capsular osmotic pressure and colloidal osmotic pressure and capsular hydrostatic pressure. So glomerular hydrostatic pressure is 60 millimoles per mercury. And blood colloidal osmotic pressure. Blood colloidal osmotic pressure is exerted in the blood plasma due to presence of plasma proteins, that is 32 millimoles per mercury. And capsular hydrostatic pressure is exerted here. That means in the Bowman's capsule, in the lumen of the Bowman's capsule. Why? In the lumen of the Bowman's capsule, already some filtrate is present. That filtrate is having some pressure. That pressure is opposing the glomerular blood pressure, that is 18 millimoles per mercury. So the 60 millimoles per mercury is opposed by 32 millimoles per mercury plus 18 millimoles per mercury. That is 50. Total it comes around 50 millimoles per mercury. So this 50 millimoles per mercury is stopping the 60 millimoles per mercury. Then what is the net filtration pressure? Net filtration pressure is glomerular hydrostatic pressure minus blood colloidal osmotic pressure plus colloidal capsular hydrostatic pressure, that is 60 minus 50 equal to 10. So the net filtration pressure is 10 millimoles per mercury. So this 10 millimoles per mercury causes the filtration of the blood. So what is the volume of blood filtered by kidneys means? The volume of blood filtered by kidneys is around 1100 to 1200 milliliters per minute. That is 1/5 of the cardiac output. So the cardiac output, you know that it is 5040 ml per minute. So next one is renal plasma flow. So if 1100 to 1200 milliliters of blood is filtered by kidneys per minute, then how much plasma is flowing through the kidneys? Or how much plasma is filtering through the kidneys per minute means it comes around 660 ml. Why? The plasma composition of plasma in the blood is 55%. So what is 55% of 1200? So if you calculate like that, you will get 660 ml. For example, you see 100% is equal to 1200, then 55% equal to how much? Okay, you will get the answer as 660 ml. Okay. Now let us see. The filtrate collected in the Bowman's capsule is called as glomerular filtrate. The glomerular filtrate is also having another name called as renal fluid, or it is also called as primary urine. Then what are the substances that are filtered and what are the substances that are not filtered in the Bowman's capsule? Substances that are filtered in the Bowman's capsule includes the water, ions, glucose, amino acids, vitamins, and enzymes. All these substances are essential substances which are useful to the body. Then there are also substances filtered. There includes ammonia, urea, uric acid, and creatine. These are waste materials, and these waste materials should be excreted outside. Substances that are not filtered includes RBC, WBC, platelets, and proteins. Because these substances are having high molecular weight, so they are not at all filtered through the filtration membrane. So the filtrate that is formed in the Bowman's capsule is called as primary urine. So what is primary urine and what is secondary urine? Let us see. The liquid collected in the Bowman's capsule is called as primary urine. Then it contains waste plus useful substances. The concentration of primary urine is hypotonic to blood. Hypotonic to blood means the concentration of primary urine is less than the concentration of blood. Then what is secondary urine? Secondary urine means it is a liquid collected at the end of collecting duct. So the liquid collected at the end of collecting duct contains only waste materials and it is hypertonic to blood. Hypertonic to blood means it is having high concentration when compared to the blood. Right. Then what about the volume of filtrate collected in the Bowman's capsule? The volume of filtrate collected in the Bowman's capsule is called as glomerular filtration rate. Okay, GFR. Then what is the GFR? GFR is 125 ml per minute. That means per minute, the glomerular filtration rate is 125 ml. Per hour, it is 125 into 60. Per day, it is 125 into 60 into 24 hours per day. So how much it is formed? It is formed into 18,000 milliliters, that is 180 liters per day. So how much of urine a person excretes per day means it is 1 to 1.5 liters per day. That is, the urine excreted is 1% of GFR. Then what is the percentage of GFR that is reabsorbed is 99%. Okay, 99% of GFR is reabsorbed back. Okay, let us go for the next one, selective reabsorption. So movement of substances from renal tubule into blood via interstitium. So this one is the renal tubule. Okay, and this one is the blood capillaries that is surrounding the renal tubule, and I'm going with the green color here. So this green color is the interstitium. Okay, interstitium means that is the tissue that is present between the nephron and the blood vessels. The tissue that is present between the nephron and blood vessel is called as interstitium. So the tissue that is present between the nephron and blood vessel is called as interstitium. So movement of substances from renal tubule into blood. From this is the renal tubule and it is moving into the substances are moving into the blood. That is called as selective reabsorption. This one is called as interstitium. So what are the substances that are moving from renal tubule into the blood through interstitium is, first one is high threshold substances. High threshold substances means these are the essential substances. They are 100% reabsorbed. See the examples, which are the essential substances: glucose, amino acids, proteins, and vitamins. All these substances, when they are filtered and enter into the lumen of Bowman's capsule, again they are reabsorbed back into the blood. Low threshold substances. These substances are absorbed in little amounts. What are they? Urea and uric acid. Why these substances are reabsorbed in little amounts means to maintain the osmotic balance. A threshold substances. These substances are not at all reabsorbed. They are actually excretory substances. Examples are creatine. Right. So let us go for the next one. Substances reabsorbed by which type of transport? Let us see. Glucose, amino acids, and sodium are reabsorbed by active transport. Nitrogenous waste materials are reabsorbed by passive transport, and water is reabsorbed by passive transport. Then the substances present in the blood and substances present in the glomerular filtrate. What are the substances present in the blood and what are the substances filtered and entering into the glomerular filtrate means glucose present in the blood is filtered. Proteins present in the blood is filtered. Amino acids present in the blood is filtered. Creatine present in the blood is filtered. So this glomerular filtrate reabsorbed. So glucose that is present in the glomerular filtrate is reabsorbed 100%. Proteins are reabsorbed 100%. Amino acids are reabsorbed 100%. From glomerular filtrate, creatine is not at all reabsorbed. So this creatine is 100% excreted through the urine. Then in urine, there is no glucose, there is no proteins, there is no amino acids also, right? Now let us go for the next one, tubular secretion. So tubular secretion is also called as augmentation. What is meant by tubular secretion? Tubular secretion means it is a movement of substances from blood into renal tubule via interstitium. So these are the substances present here. These are substances are moving from blood through the interstitium into renal tubule. So this one is the interstitium. So this is called as tubular secretion. What is the use of tubular secretion means to maintain ionic balance of the blood or to maintain pH balance of the blood. What are the substances that undergo tubular secretion means H+ ion, K+ ions, ammonia, pippuric acid, oxalates, and creatinine. All these substances are reabsorbed from blood into renal tubule to maintain the pH or to maintain the acid-base balance or to maintain the ionic balance of the blood. Right. Then functioning of tubules. Let us go for how this urine is formed. Let us see now the formation of urine in the PCT. So what are the substances that undergo selective reabsorption? What is the substances that are secreted into the PCT? So PCT is the main site of selective reabsorption. Very important point. PCT is the main site of selective reabsorption, and obligatory water reabsorption occurs. Obligatory means compulsory. So compulsory water reabsorption occurs in the PCT. Okay. So what are the substances reabsorbed means? 70 to 80% of substance water is reabsorbed. 70 to 80% of electrolytes are reabsorbed. 100% glucose is reabsorbed. 100% amino acids are reabsorbed. 100% proteins, vitamins, and hormones and enzymes are reabsorbed. Then what are the substances that are secreted into the PCT means? H plus ion, K plus ion, and ammonia. So the concentration of renal fluid in PCT is isotonic to blood and cortical fluid. So you know that PCT is present in the cortex of kidney. The concentration in the PCT is equal to the concentration of blood. Okay. If the renal tubule is removed from the nephron, urine is more diluted. So urine is more diluted. So obligatory water reabsorption occurs in the PCT. So this completes about the functioning of tubules in the PCT. Let us go for the next one in the next class.